Hello and welcome to the last lecture in this series of lectures on chemistry in everyday life. I hope you have had a good time so far in the last 3 lectures. In today's lecture, we will study about food additives, and in food additives, we will study specifically about artificial sweeteners. Then we will move on to antioxidants and the role of antioxidants. Eventually, we will discuss food colorants and finally, chemistry of cleansing agents. So, let us begin. Food additives. What are food additives? Any chemical or ingredients which are added to a food for several purposes, and those purposes would include let us say improving upon the taste of a food. They can also be added to preserve the flavor of a food. Then sometimes it is just to increase or enhance appearance of a food, right. Let us say an ice cream on a strawberry flavor should be, you always expect it to be bright red colored like strawberry, right. Any dull colored ice cream might not give you that kind of appeasement. And sometimes it could be to enrich nutritional value of a food, right. So, with this there are various kind of food additives, and this would include food colors. This would also include flour improvers and also sweeteners, antioxidants, preservatives, fat emulsifiers and stabilizing agents, the kind of molecules you always use for those ice cream preparations and all, right. Preservatives you use while preparing those pickles and that kind of stuff when food has to be kept for a long time. And then certain nutritional supplements such as minerals, vitamins and amino acids. So, nowadays this has been a trend of having those high nutritional value those syrups or those drinks which are abundantly available in market. For this particular lecture, which happens to be the last lecture in this series, we will be discussing about sweeteners and antioxidants, including food colors. So, these 3 types of things molecules we will consider in these food additives. We will begin our studies with food sweeteners. So, sweeteners can be basically of 2 kinds. One type of sweeteners are the ones which are obtained from nature, right. So, something like sugar and or maybe honey. When we talk of sugar refined, it is not exactly natural, it is quite refined, been processed. But let us say you look at those jaggery for example, that is more natural in its comparison. Then there is an entirely different class of compounds which is man-made and artificial. Nevertheless, they are also very sweet, and these are a class of artificial sweeteners. Now, example as is being shown in this slide, in this picture, is a very famous artificial sweetener called saccharin, right. Now, there is another thing. Most of these natural sweeteners are generally of high nutritional value. You may not call it great, but certainly they are high nutritional value, meaning thereby they provide you enough energy, right. For example, you take some sugar and you get glucose within no time, right. And on the other side, well, when we talk about artificial sweeteners, generally these sweeteners they would have a very limited nutritional value, and sometimes they would have no nutritional value, which means that for most of these molecules the body is not able to process these molecules in any way which will provide energy to the system, right. So, no glucose, nothing. So, in order for any body any molecule to have a nutritional value, it must be able to produce those ATPs, must be able to somehow find its way into this Krebs cycle and produce energy. So, these molecules are not able to work in that manner. As far as their artificiality is concerned, not all the molecules having sweetening properties with no nutritional value are artificial. There are some compounds like steviosides, etc. They are actually found in nature, and they won't have any nutritional value, but still they are extremely sweet compounds. You have read so much about sugars, so we are not going to discuss anything about natural sugars. The kind of molecules which interest us are these artificially derived non-nutritional sweeteners. Okay. Why do we need these kind of non-nutritional sweeteners? Primarily for 2 or 3 reasons, as you could also probably assume. First one being for patients who are suffering from diabetic, right, because in diabetic you really would like to control your insulin machinery, and in order to control insulin machinery you don't really would like to feed it with ready-made glucose molecules, right, something of the kind of sugars when we intake. So then diabetic patients are generally refrained from taking sugar rich food, leave alone sugars. So for these molecules, for these patients to get a flavor of sweetness, it is important that they thrive upon certain other class of compounds, and these non-nutritional sugars, non-nutritional sweeteners, they would come handy for these patients. Second is the kind of people who are suffering from obesity and they would like to reduce waste. Now, one good way to reduce waste is you don't thrive upon those sugars. So again, for such people, in order to have that tinge of sweetness in foods, they would rather like to go on non-nutritional foods than to thrive upon the traditional sugars, right. Another very important thing is about the economics. When you talk of most of these artificial compounds, one thing is invariably seen in these compounds that they are extremely potent on a sweetness to weight basis when compared to sucrose, which is your natural sugar, right. Let me elaborate on this point. In the coming slides, you will come to know what I really mean by this. So these are some of the molecules we are going to study, 6 basic class of compounds. We will discuss aspartame, we will discuss saccharin, sucralose, acesulfame, cyclamate and allitame. These are all approved sweeteners in most of the countries, and as and when I'll also show you in certain countries when some of these molecules or one of these molecules are not used, I'll show you the examples as and when we discuss them. So the important point to mention here is how much sweet is an artificial sweetener, which means how would we measure a sweetness potency of a particular molecule, right. For example, we generally claim that aspartame is 200 times more sweeter than a simple sugar molecule, which is sucrose. Okay. So it is 200 times sweeter. Now the point is, how do we measure it? Well, that's very interesting. You would be surprised to know there is no instrument which would measure sweetness for you. Instead, there are a set of experts, trained experts, which are human beings, people, right. They are trained expert people who would tell you relative sweetness of a particular compound relative to 5% sucrose solution or 10% sucrose solution on a scale. So let's see, they are first trained hard to identify sweetness of 5% sucrose solution or 10% sucrose solution, all right. And then the molecules which are to be evaluated for sweetness are fed to these experts. For example, let us say 0.025% of aspartame is equivalent in sweetness to 5% sucrose. Okay, 5%, they feel like this type of sweetness which gets by having 5% sucrose is equal to aspartame which is having a consistency of 0.025%. In this case, potency of this aspartame is 5 upon 0.025, which turns out to be 200. which means potency of aspartame is 200 times more than sucrose. So, isn't it interesting, right? So, more interesting thing is coming just now. Now, very interesting thing, response versus concentration curve here, or this graph here for sweetness, which means if you keep on increasing concentration of a particular sweetness, what is the response? So, for example, if 5% solution will give you this much sweetness, okay, to this point, then let us say 10% of that solution will give you a response which should be just double, right, and then 20% should be double of 10%, right. But then that is not the way it happens. So, you do not get this kind of a linear response. Instead, like you see in this example, 2% sucrose solution would have a potency of 625 relative to aspartame, okay, which means aspartame is 625 times more sweeter in comparison to 2% sucrose solution, right. But when you take this measurement relative to 10% sucrose solution, potency decreases to just 110, right. So, what you would generally note is increasing the concentration would no longer produce increasing sweetness, and then a response would asymptotically approaches a maximum value. This a maxima is approached in no time. That is a very commonsensical response to draw, right, because what would happen, since there are experts, there is a human intervention which is going to measure sweetness. So, beyond a certain point, every sweet will be very sweet or extreme sweet. It will be limitation of our own ability to judge, okay. Our receptors, our sweetness receptors, they will be completely saturated with those molecules, and it will be very difficult for a person to judge the relative sweetness of two molecules, okay. Now, I already mentioned about the receptors, right. So, when we talk of mode of action of these sweetener molecules, it means the second question we are coming to is how do our body perceives a molecule or some taste as sweet? That is actually a very difficult business for scientists to do, simply because you must understand taste cells actually lies in this epithelia, which epithelia is actually surrounding these taste buds, right. And these epithelia, these taste active cells are very difficult to isolate. More difficult is the phenomena of actually isolating exact receptors which are which are actually able to send a signal for sweetness. And in the previous lectures, I have already apprised you about the receptors and how receptors behave. So, the problem in hand here is that to isolate receptor specific binding of those sweetener molecules from non-specific binding to other components is extremely difficult, which means that people have gathered this opinion that these sugar molecules and other non-sugar sweeteners, they bind to receptors also, and apart from the receptors there are also certain non-receptor areas where these molecules can bind and elicit a sweet response, okay. What has been known so far in this area is this. So, this is a particular cell, a taste cell, okay, which is to elicit this sweet response here. Basically, giving you sweet responses to kind of give a signal to adjoining nerves, and a signal to passing nerves will be given through opening and closing of these ion channels. In case of sugars, the ion channels in question are potassium and calcium ion channels. So, closing of one ion channel and opening of another ion channel will give you enough response to the adjoining nerves, which will take the signal to brain and make brain feel that something sweet has been taken, okay. Now, manipulating these ion channels can be done in two ways. A molecule can directly bind to these ion channels, right? In the other case, molecules can actually bind to a particular receptor, and through binding of this receptor, a cascade of activity will start to happen. And because of this activity, what happens is basically certain enzymes are activated. Because of those enzyme activation, certain secondary messengers are evolved, and those secondary messengers are able to finally control this type of ion channels here and elicit a sweet response. It is broadly perceived amongst the scientific community that non-sugar sweeteners or non-nutritional sweeteners, they generally bind to these type of receptors, whereas sugar molecules, those sweeteners, those sucrose, glucose, other molecules, they generally bind to these type of ion channels. Okay? And this is actually one reason why these non-nutritional sweet molecules are actually so sweet, because a very small amount of receptor being impregnated can elicit a huge response, and that we have seen during our drug design lectures, previous 2 lectures or so, right. So, with this I will now take up all those examples one by one and apprise you of the merits and demerits, if any, of these non-nutritional sweet molecules. The first one in hand is aspartame. Aspartame, as you could see, is a dipeptide, and it is a dipeptide of aspartic acid and phenylalanine ester. So, if you note here, this part is aspartic acid, right, this part, and this part is a methyl ester of phenylalanine. I hope you are aware of these these amino acid residues. Now, this molecule is around 200 times more sweeter than table sugar. Hence, a very small minute amount is consumed, resulting in negligible calorie contribution, since it is not entirely non-nutritional. It provides 4 kilocalories per gram. The discoverer of this compound was James Slater in 1965, and by 1981 FDA approved its use as a sweetener molecule. As the legend says, this scientist was actually working on anti-ulcer compounds, and just as serendipitous ways, he just licked his finger and he found out extreme sweetness, and that is how he zeroed on to this particular compound. This compound aspartame is used in almost all food categories, but major consumption is in beverages. Now, if you look at synthesis of this molecule, as organic chemist, I think that should interest you. You have to combine those two peptides, those two amino acids, right. So, you begin your synthesis with aspartic acid here, and this aspartic acid, this amino group is actually protected. We would not like to give more consideration on to these protective groups here, but for your understanding, you must keep in mind that this is protected. Now, you treat it with acetic anhydride, and by treating with acetic anhydride, this will result in a formation of an anhydride here. So, you form an anhydride of aspartic acid. Now, with this anhydride, it requires just a nucleophile to probably act upon any of these two carbons. So, you take phenylalanine, and a methyl ester of phenylalanine is reacted with this, and this phenylalanine molecule will act on this carbon to give you this type of a dipeptide, a protected dipeptide, and eventually this protection is finally removed using reductive conditions, and one gets this aspartame molecule. Now, there are couple of questions and very interesting observations which I must share with you. Generally, in formation of peptide bonds, it is a commonplace knowledge to activate your carboxylic group, right, and that means you would have to incur an additional synthetic step. Now, in this case, the one who probably created its synthesis had been very meticulous using acetic anhydride here in order to just activate this compound into an anhydride. So, there is no need of any activation reagent here in this particular reaction. The second thing about this is, as I told you, this nucleophile in amine will act on this particular carbon and the resulting molecule will be aspartame. With equal propensity, this amine will also can also react on this carbon and give you a side product, which will not be aspartame, right. So, this is something which one would need to be wary of in this particular synthetic method. So, there is a propensity of a side product in good proportions which can form out of this reaction. Another important thing, once you take aspartame molecule, once you take aspartame as a sweetener, it tends to get metabolized in the body. Now, I told you that all these are non-nutritional. It means they are not going to find their way in Krebs cycle. In fact, whenever you take any of these non-nutritional molecules, body will recognize them as foreign particles and the body will like to get rid of them as soon as possible, just like a drug molecule. Okay? So, they will find the same way as a drug is eliminated from the body, which means it will be metabolized in liver as soon as possible. So, the moment it reaches liver, it will be acted upon by enzymes and it will be hydrolyzed. This peptide bond will be hydrolyzed, resulting in formation of both the monomers, which is aspartic acid and phenylalanine. Now, the problem is for people who suffer from a disease called phenylketonuria, which is their body's inability to process phenylalanine, and then phenylalanine tends to get accumulated in the body. So, for such patients, using aspartame as a non-nutritional sweetener is certainly not advisable, right. So, let us move on to the second molecule. The second molecule we are going to study is saccharin. This is the most widely used non-nutritive sweetener in the world. Molecule is benzene isothiazole dioxide. So, it is an isothiazole dioxide here. This molecule is sparingly soluble in water, hence it is used as a sodium and calcium salt. Because there are metals involved here, this does not provide a very sweet, clean sweet taste, and generally one is left with a small, a minor feeble bitter or metallic of taste after some time. Discoverer of this molecule were Ramson and Fahlberg. They were working on making food preservatives from coal tar, wherein one of those scientists spilled a bit of the sample on his hand and observed extraordinary sweetness. This molecule is 300 times more sweeter than sucrose or sugar, and therefore, it is more sweeter than the first molecule we studied, aspartame, right. Second thing is, it is cheaper than sucrose on a cost per sweetness basis. So, that I told you, since you require such a small amount of a compound, their cost becomes even smaller, right, on a relative scale. So, if you look at the synthesis of this molecule, again synthesis is very simple and conform to the standards to which you students are exposed in your 11th and 12th. So, we start with methyl anthranilate. Basically, it is an anthranilic acid. So, a methyl ester of anthranilic acid is the starting compound for this reaction. Now, this substrate is acted upon with sodium nitrite and some sulfuric acid in very low temperatures. So, you know which reaction is going to occur. I am talking about diazotization reaction, and as a result of diazotization reaction, a disonium compound will form. Now, this disonium compound, as it is such a wonderful electrophile, we impregnate it with nucleophiles in sulfur dioxide and chlorine using a process called Momi process. As a result of this process, one gets this sulfoxyl chloro derivative of this anthranilic acid. Now, once you obtain this compound, it is treated with amine, with ammonia, and what ammonia does is it acts as some sort of bi-electrophile here. So, it replaces this ester group from this carbon and at the same time it replaces this chloro from this sulfuryl chloride, and eventually one gets with the final compound saccharin. It has good chemical stability and very low cost, and therefore, it is used, as I told in the previous slide also, it is the most widely used sweetener. It is used in toothpaste, it is used in mouthwashes, it is used in pharmaceuticals, in foods and beverages, in all plethora of activities. Okay. There are certain safety concerns with saccharin, though, since there had been certain reports of its being associated with bladder cancer, although it's not been really confirmed, and this part is still debated. But so there is some small caveat with which one should actually take these kind of molecules. With this, I come to the next molecule in line, which is acesulfame K, and K denotes potassium here, since it's a salt of potassium. It's a member of dihydroxythiazanone dioxide. So again, you have this sulfur dioxide moiety here, and it is an oxathiazone. So you have this oxathiazone here. This is a very highly stable crystalline sweetener. It is stable to hydrolysis even in highly acidic beverages. It has extreme water solubilities. It was discovered by Kloss and Jensen in 1970. Now, if you look at the its sweet index, its sweetening index, you find that this compound is 200 times more sweeter than sugar. And the other important point about this is it is perceived quickly without any unpleasant delay, as we saw with the previous compound we studied, saccharin, right. Now, again, if you look at the metabolites, again this is a foreign particle, so body would like to get rid of this molecule. The advantage is, if you look at the metabolite, once it is acted upon hydrolytically, one gets this ring opened up into a bond cleavage occurring at this sulfur oxygen bond. One gets this kind of acetoacetamide sulfonic acid, and this compound is a physiologically benign substance. It has not shown any toxicities, hence this compound you can consider as a safe compound to intake as a replacement of sugar molecules. So, as far as synthesis of this compound acesulfame K is concerned, you start with an isocyanate. Isocyanates are these molecules with this functionalities. C carbon joined with an oxygen and joined with nitrogen through double bonds. So, this makes this carbon extremely electrophilic. Okay. So, what we do is it is treated with acetone, and with acetone it is with this carbon acts as a nucleophile here, acts on this carbon and eventually gets to this particular compound, which is halosulfonyl acetoacetamide. Now, if you treat this again with a base, what base is going to do is abstract a proton from this position, this methyl position, and eventually convert this structure into an enolate, right. So, you would have a double bond here and OH, and you would have a double bond here and CH2. So, with this enolate, there is a possibility of nucleophilic attack from this carbon as well as from this oxygen to this sulfur, since one would like to get rid, this compound would like to get rid of this chlorine here, right. It is a intramolecular cyclization reaction. What eventually happens is it is an oxygen which reacts, and basically you can ascribe this reaction to hard interactions. You have oxygen as an hard anion here, and you have sulfur as an hard electrophile. So, if you look at applications of this, it is a non-caloric sweetener. It is excreted completely and rapidly in urine. It is used in low calorie and calorie reduced beverages. Generally, it is blended with other sweeteners, and generally those synergistic effects of sweeteners are they give a better quality for these artificial sweeteners. Next molecule in line is cyclamate. Cyclamate is a salt of cyclamic acid, and cyclamic acid is this compound, this sulfonic acid here. So, a sodium salt of this sulfonic acid would be considered sodium cyclamate. If this is calcium salt, then it is called calcium cyclamate. So, generally people who are on low sodium diet, they are given this calcium salt, and otherwise sodium salt is the one which is prevailed more. Discoverer of this compound was Michael Sweda in 1937. If you look at the relative sweetener index, you find this compound as 30 times more sweet in comparison to sucrose. There is again some bitter off taste, but it has good sweetness synergy with saccharin, and it is stable over wide range of pH and temperatures. Synthesis of this is quite straightforward. You start from this cyclohexylamine, and sulfonation of this cyclohexylamine under basic conditions will provide you with this sulfonyl derivative of this hexylamine, which is nothing but cyclamate. Likewise, if in order to metabolize this compound within the body, it will again get converted into cyclohexylamine, but the problem is this cyclohexylamine has been found to bring chromosomal damage in animals and tumors in rats. So, it is not approved for sale by FDA, and it is also not used in Indian markets just for the same reason. So, it is considered a carcinogenic compound. Another compound is sucralose, which is just a trichloro-derivative sucrose molecule. So, you note a chlorine here, another chlorine here, and you have another chlorine here. Right now, it is approved in Canada, Australia, and recently it has been approved in US also for sweetener use. So, there were these scientists Tete and Lyle. They found that substitution of hydroxyl group of sucrose by halogens, it was found to increase sweetness potency dramatically. So, what they did was they converted this sucrose into trichloro derivative, which is sucralose, and it has a pleasant sweet taste. It is stable over wide range. However, as you would also note that discolorization, and there would be some amount of HCl liberation, hydrochloric acid liberation with this molecule at high temperatures. Hence, one should be careful of using this compound also. I am not going into the synthesis of this molecule now, simply because it involves a lot of protection and deprotection steps, which I do not consider in purview of this course. So, this brings me to another dipeptide, which is allitame, and if you note here, it also contains aspartic acid and alanine, and with this there is another component which is tetramethylthioetane. Okay. This compound is extremely sweet. So, it is 2000 times sweeter than allitame. It is much more stable also than aspartame. Again, I am not going into nitty-gritties of synthesis of this molecule as well, since it also involves a multi-step process, which I feel should not be in the purview of this discussion. So, broadly what we look into is whenever you look into a molecule as an artificial sweetener, there are certain properties. It should be cost effective, it should have improved properties, improved in terms of less toxicity, no side effects and easy metabolism, and then it should also have a viable process for commercial production, as we saw in case of aspartame, saccharins. They are very easy processes, or cyclamates and all. So, easy reactions, easy synthesis, cost effective methods and better properties are the ones which people look for, and this is still a burgeoning area. Many people are continuously working on this area to find out new molecules with better properties as sweeteners. Okay. So, with this I will take this journey forward to next kind of compounds, which are antioxidants, and basically you must understand why one would require antioxidants, because in our body during normal respiration process, during normal digestive processes, whenever we take some food, there is always generation of certain free radicals. Okay. Even with simple oxygen which we take for breathing gets converted into kind of oxygen free radical, which converts into peroxy free radical, and that free radical, peroxy free radical, can actually go on and form hydrogen peroxide in the body. So, these are called reactive oxygen species which keep on getting formed as as a side product, you can say, of metabolic processes taking place in the body. Now, the problem is that even if they are side processes, they are extremely hazardous, okay? And they are hazardous to an extent that these peroxides tend to react on and tend to act upon cellular membranes. They act on lipids present on cellular membranes, they act on nucleic acids in DNAs, they act on let us say other protein structures which could be part of very important enzymes in the body. So, gradually, slowly and steadily, they tend to kill these structures, okay, tend to distort these structures, and with distorting of these important biomolecules, longevity of a human being tends to decrease. So, then that is why we say a person having a lot of free radicals probably will have a lot of disastrous or negative processes going inside. So, now there comes this class of antioxidants molecules which are there to scavenge these free radicals formed within the body, right. So, antioxidants, they act as scavengers, and they scavenge peroxy and oxy free radicals formed during auto-oxidation radical chains. So, I told you there are these auto-oxidation processes continuously taking place within the body even as you breathe. So, what basically happens when these peroxy radicals, for example, they tend to generate reactive free radicals from biomolecules, any of them could be represented as this R free radical. So, these free radicals then tend to engage in chain propagation steps, meaning thereby they will constantly react with oxygens, produce these peroxy radicals, and these peroxy radicals will again generate this R here, this R free radical, forming this peroxy acid in process, right. Now, what would an antioxidant do then? Antioxidants have this inherent ability to kind of generate free radicals which are able to stop these chain propagations as and when these happen. So, mostly these antioxidant molecules, they contain certain groups, generally phenolic groups, which play a major role in stabilizing these type of these type of free radical compounds. So, they stop this chain propagation from occurring forward, and that is how they stop this oxidative processes occurring in the body called oxidative stress within the body. Okay. We will discuss only one representative compound, and this compound is vitamin E, which is more specifically called tocopherol. It is found in plants and animals, and it is a necessary complement of antioxidants, right. There are other vitamins also like ascorbic acid. Vitamin C is also a fantastic antioxidant molecule. We will only consider this vitamin E. Generally, lipids on the surface of cereal food are easily oxidized and emit unpleasant odors. You might have seen that and heard about a process called rancidity. So, when you leave a fatty acid, when you leave an oil for a long time in oxidation under oxidative atmosphere in air, you would see that after some time it will start to give you a very foul smell, and that smell is that process is called rancidity. That smell comes out of those peroxy acids which are formed. So, in order to preserve those oils, generally vitamin E is added or sprayed onto these molecules. So, what vitamin E in fact does, I told you this process of generation of these peroxy radicals, right. So, you look at this molecule now, this is a molecule of tocopherol, and because of this oxygen here, it tends to get oxidized through a single electron transfer reaction, forms a free radical here, and in the process of forming a free radical, it actually is able to provide another electron to this peroxy radical and stop this propagation process right here, and in turn it itself gets stabilized into formation of this kind of dimeric species, or sometimes some molecules can also engage in formation of quinones. Okay. So, in that case they are able to release 2 electrons, 1 electron at a time, right, and all those quinones or these kind of dimeric species are very stable compounds. So, they do not go any further oxidative processes, right, and that is how they put a full stop to an oxidative process occurring within the body. Coming to the next class of studies is food colorants. Again, in this case food colorants could be natural pigments, they could be added colorants. I will keep my discussion to natural pigments, wherein I will discuss about carotenoids, which are responsible for colors in various vegetable products and in animal products like fruits, eggs, dairy products and cereals. If you look at the structure of these carotenes, you would note that these are highly conjugated structures, right. Look at the double bonds in conjugations. There are so many double bonds in conjugation. It has a logic. A molecule having extended double bond, conjugated double bonds tend to be a colored compound, simply because what would a compound need to be colored, right. If you understand this, you know, a compound, in order to behave as a colored compound, should be able to absorb photoelectric radiation. So, those photo radiations, those photons, right. Now, with extended conjugation, difference between the filled orbitals of these molecules and unfilled orbitals, the difference starts to become lesser and lesser, and there is a point that it becomes so less that a small frequency, a small energy photon coming from photo irradiation is enough to make that electron jump from an unfilled orbital to a filled orbital. It means it has started to absorb radiations, and through absorption it will give you a certain color. So, that is a reason why you need extended conjugation in molecules in order for them to behave as colored. Another class of compounds is anthocyanins, which are, again, if you look at, there is an extended conjugation in the molecules and polyhydroxy functionalities, and this provide you all those bright colors seen in berries, in different other fruits. One interesting thing about them is, generally if you have more hydroxy compounds, the color shifts from red to blue. Okay. At the same time, if you have more of methyl substituted by methoxy, again the color changes from blue to red. So, you look at these examples here, palerogonadin, cyanidin, delphinidin, and you see in strawberry you have palerogonadin and cyanidin. So, the color is red. In case of delphinidin, cyanidin, in black currants, the color is more in this blue hinge. Okay. This brings me to the last topic of today's lecture, and that is about cleansing agents. Cleansing agents is, you all know water is a fantastic cleansing agent, right, since you do not need anything to clean your hands other than water. But sometimes water is not good enough. So, you need to wash your hands with soaps and detergents, which are all class of synthetic cleansing agents then. Why water is not good? Because there are dust particles which water itself is not really able to take out, and oily particles, greasy particles, which water alone is not able to take out from the skin or from clothes, and hence one needs soaps and detergents. So, what are these soaps? These soaps are basically sodium or potassium salts of long fatty acids. You know about fatty acids, right? So, a sodium salt or potassium salt of a fatty acid will be generally will be a soap of different kinds. So, for example, in example here, this is a soap of stearic acid, this is a soap of palmitic acid. If you know how these fatty acids are produced in plants, you would understand that they are always produced as triglycerides, which are called fats. So, a fat is a triglyceride molecule of a representative fatty acid. So, if you react this fat with a strong base like sodium hydroxide and heat it, you would find that hydrolytic action of base will take place, and this hydrolytic action of removing this ester from this glycerol molecule is called saponification. So, as an act of saponification, one gets a soap molecule and glycerol. Glycerol can be retrieved back, and that is how one gets soap. There are various kinds of soap: transparent soaps, medicated soaps, laundry soaps, scoring soaps, shaving soaps, etc., depending upon the kind of the use for which they are made, and that is done by using certain other additives like adding perfumes to soap, adding certain medicinal compounds to soap, so on and so forth. What is the phenomena of a soap taking dust away? Look at this surface. This is a surface of a cloth, for example, right. This surface contains a lot of grease. Okay. Let us say these oily particles are on the surface of a cloth. Now, when you add water, when you add soap to it, soap has a fat loving tail and a water loving head. So, once in water, it arranges in this type of globular or round structures in which the core is surrounded by these fat loving tails, whereas the surface is surrounded by these polar chains. So, within this core, they actually engulf this fat particle or this oil or this grease, and basically this grease is separated into emulsions, and hence now when you wash it vigorously with water, water is able to carry out these emulsions, take away these emulsions, leaving this surface of the cloth clean for you. Problem and limitation with soap is, if you treat it with hard water, a water which contains calcium, water which contains magnesium or iron, they tend to form salts of magnesium, calcium or iron, and all these salts, they form insoluble precipitates, and these insoluble precipitates then render activity of soap ineffective. In order to address this, one actually resort to detergents which have stronger cleaning action than soap, which works better in soft and hard water both. So, if you look at their composition, detergents composition, you generally see they are anionic, they can be cationic or they can be non-ionic. So, as the name suggests, we will consider first anionic detergents, which means that this time the long chain has to be anionic. So, one way to get to this anionic chain is sulfonation of a long fatty acids. Now, in this case, fatty acid is first reduced to an alcohol. For example, in this case you have lauryl alcohol, and this lauryl alcohol is sulfonated using concentrated sulfuric acid, which gives lauryl hydrogen sulfate, and then this sulfate is hydrogen sulfate is just reacted with a base. So, in order to get a requisite counter ion. So, in this case, this whole fatty acid chain is anionic with a counter ion which is cationic, and this is a class of anionic detergent. Sometimes you really need not natural fatty acid, use those natural fatty acids. So, in this case one can start with dodecyl benzene. Again the same way, this is being sulfonated at one end of this benzene, and eventually a counter ion is generated using treatment with sodium hydroxide, and one gets sodium dodecyl benzene sulfonate. Again that is an example of anionic detergents. So, look at the properties of and uses of these anionic detergents. First of all, they are cheap to manufacture. As we just saw, what you need is just convert them, if you are talking about a natural fatty acids, just reduce it to an alcohol, and that alcohol can actually be sulfonated, and that sulfonated can provide you an anionic detergent. So, they are cheap to manufacture. Second, petroleum industry provide a lot of raw materials for this, as we saw in the previous examples. They are very efficient in cleansing action, and they are generally commonly used in household works. Okay. So, the same thing about cationic detergents. In this case, the long chain has to be cationic now, and a counter ion should be anionic. So, one easy way to get a cationic long chain is to use quaternary ammonium salts. So, one takes quaternary ammonium salt here of cetyl trimethyl compound. So, you have this long chain and a terminal quaternary nitrogen bearing a positive charge, and the salt of it is with a halogen, a bromine here will give you a cationic detergent. Now, in comparison to anionic detergents, cationic detergents have poor detergency. They are used for germicides or fabric softeners and specialist emulsifiers. Now, important point to note is they are generally expensive in comparison to other anionic and non-ionic detergents, and they are very less used. They have limited applications, as you can also see. Now, as far as non-ionic detergents are concerned, now in this case you do not need ions, and the an easy way to go around it would be just to take a fatty acid or an acid and react it with an alcohol. This time this alcohol should be different from glycerol as nature does. So, one gets an ester of this compound. In this particular example, we have taken stearic acid, it is reacted with polyethylene glycol, and one gets a compound which is this this ester here, which is non-ionic detergent. Okay. As far as properties and uses of non-ionic detergents is concerned, they have enormous properties. They enormously they are used. They are the biggest group of detergents. For example, in liquid dishwashing detergents, they are generally used, and other applications also. Now, one important point I told you about detergents is that they can be used with both hard water and the normal water. So, what happens when they are treated with hard water containing salts of magnesium and calcium? So, in this case I am taking an example of anionic detergent, and this anionic detergent upon reaction with hard water using these having these salts of magnesium will form a magnesium salt of this sulfonate. But unlike magnesium salt in case of soaps, magnesium salt of those fatty acids which I told you were not soluble in water, in this case these magnesium salts of sulfonate, since sulfonate is very polar. So, this salt is going to remain soluble in water, and therefore, it does not interfere in its cleaning properties of the detergent, and that is a big advantage with detergents. But again, with every good thing it comes with certain caveats. So, when you talk of detergents, there are also certain disadvantages, and an important disadvantage is their being non-biodegradable, and their being harmful to skin also to an extent. More than that, they tend to produce stable and harmful foams in water, which creates soil pollution, which creates danger to aquatic life, which also does water pollution at large. So, one must also look into alternatives, into other compounds in future. This is also a science which needs to be taken more seriously, and other compounds need to come with detergent actions which should have which should be easily metabolizable, which should be biodegradable, and should be easy on mother nature. Okay. So, with this our topic is coming to an end. Before I finally close down, let me summarize what we did for this particular lecture. We started with food additives, and in food additives we specifically targeted sweeteners, antioxidants and food colors. In sweeteners, I spoke about all the prevalent molecules present in the market as artificial sweeteners, their their limitations, their uses and their usefulness. In antioxidants, we limited our discussion to molecules from nature, okay, which impart colors to foods and other things. Then finally, we moved to cleansing agents. In cleansing agents, we discussed soaps and detergents, advantages of detergents vis-a-vis soaps, and also their limitations and their impact on environment. This lecture was prepared using these references shown on the screen, and students are encouraged to go to these references in order to further read and inquire about this topic. With this, I thank you all for your patient hearing, and this was the fourth lecture and last lecture in this series. So, I hope you must have drawn some very important points from all these 4 lectures. Thank you very much for staying with me.